Thank you. Thank you for introduction. Can you hear me? Okay, so first of all, I would like to express my sincere thanks to organizers, especially she's not here, but very much appreciate Reiko Kuroda. She gave me a chance to talk here. I think I try to explain something like a full throat by our research, a little bit old research results, but surely scientific. I little bit worried about in case you don't know so much about technical terms, then I'm afraid you will miss. But in such case, please show me very bad face or angry face, and then I will find someone. Several people has couldn't understand, cannot understand, and then I will change my pace or word different ways if possible. So I'm personally interested in the molecular mechanism, how education and experiences in development influence to our cognitive ability and personality. For such studies, for example, we have to understand the ability to study structure of membrane proteins. For example, you'll see this is apple. And when you feel tasty and try to eat, then you will come too close to the apple and take apple and bite. But for the aim, we have to understand, for example, visual system and also muscle control, neural mass control. So, for example, structure of rhodopsin is crucially important to understand photoreception in the eye, especially at rod cell. And also acetylcholine receptor you will see here and exists actually in here is a key molecule on signal transduction for regulate our muscle, means neuromuscular junction. Then we can move. For structure analysis, one of good candidate might be electron crystallography. Show this. I will not skip. I will skip this in detail. But electron crystallography is good candidate because of following several reasons. Structure can be analyzed in membrane, means lipid bilayer. You will see the cell. Cell surface is covered by membrane. So we can analyze structure in lipid bilayer, and structure can be analyzed even if we couldn't grow very best crystal, but somehow crystal, and then we can attack structure analysis by such poor crystal. And also, very important thing is both sides of the crystal have an open space, then we have less artificial influence in three-dimensional crystal. So we can avoid such artificial interaction, and we will be able to analyze structure at a relatively native condition, even surface structure. And also phase is a little bit difficult to understand, but phase can be calculated directly from image, electron micrograph. For taking high resolution micrograph, when I start to study electron microscopy, researchers in this field argued whether electron microscopy can give anatomic image or not. Then we have to start confirm whether electron microscopy enables us to take an atomic image by using chlorinated kappa phthalocyanine. Kappa atom, porphyrin ring, and 16 chlorine atoms are clearly resolved in this image. But when I try to take this just one image, I struggled and did many non-scientific things. Because when we try to take high-resolution image, we have to adjust very accurately best focus condition, even though small focus change dramatically changes the image feature, as you will see here. This is exactly the same molecules image, but completely different. But when we try to adjust focus, using electron beam give us severe damage, destroy at all. Then impossible to take high resolution image, something like this. To overcome these difficulties, we developed so-called minimum dose system I named, minimum dose system. By using this minimum dose system, we could easily take or reproducibly take very best focus condition radiation sensitive organic molecules such as silver TCNQ complex, as you'll see. But when we move on to, that is my aim, to observe biological sample. For example, ice-embedded bacteriophage T4, it's when illuminated electron beam easily give a damage and gone. So something like this. So after long struggling trials, we carefully measured it turned out radiation damage is reduced to 1/10 and 1/20 at the specimen temperature below 20 Kelvin and 8 Kelvin, respectively, compared with the damage at room temperature. Then we started to develop a cryoelectron microscope in 1983, and we perfected the superfluid helium stage to be built into the conventional transmission electron microscope in 1986. From then, we made quite a bit of effort to improve resolution as well as operational difficulties. Now we are developing 8th generation cryo microscope for image-based structure analysis, by which we can analyze three-dimensional structure of lipid molecules in lipid bilayer without crystal. So today I will tell you all crystallography result by crystallography. But now our trend is move on to non-crystallographic technique, say single particle analysis. But I will not have time to tell very new situation. But by using third generation cryoelectron microscope, we could analyze structure of bacterial rhodopsin. That was analyzed our pioneers, Richard Henderson and Nigel Unwin, in 1975, as you will see. By using our little bit powerful cryoelectron microscope, we could improve resolution from here to here. And you will see by our structure, we can analyze three-dimensional structure of bacterial rhodopsin as well as lipid molecules. Bacterial rhodopsin is very effective photo-driven proton pump. And then proton has to fly into very narrow entrance, say smaller than 5 angstrom. The synchronized manner absorb on photon to a retinal, proton have to come into the entrance. It's almost hopeless. It's difficult. then how to do bacterial rhodopsin actually do? We analyze structure and form a trimer, and at the cytoplasmic side, trimer inside is red color I showed, means negatively charged surface. Negatively charged surface absorb proton, and proton can surface migrate freely or very rapidly, and then synchronized way of absorption on the retinal photon absorption, proton can come into entrance, even very narrow entrance, very smoothly and effectively come into, then bacterial rhodopsin can effectively pump proton. Well, in our brain, neural cell is very important. So ion channel is the key molecule on signal transduction of neural system. But at the same time, the most abundant element is water. And in our brain, water content is 80% or higher. Then we have to think about water molecules in our brain when we try to understand brain function. For example, when a native speaker speaks English, central part of cerebellum activated. But in case of Japanese speaks English, in my case, you will hear my poor English, but my brain might be activated at the peripheral part, not central part, completely different part. And then, in neural cell is consumes a large amount of oxygen, glucose, and the rising up temperature. Then for brain function, blood flow regulation is crucially important. But very strangely, or it's not naturally, vascular capillary in our brain, vascular capillary, kekkan, vascular capillary has no vascular smooth muscle. Then there is no regulation system to expand or shrink of vascular capillary. How to regulate blood-brain barrier according with our neural activation? Then we see the astrocytes surround the vascular capillary, and huge number of water channel is expressed at the end feet here. So I think if the astrocyte change the volume and or shape, then blood flow could be regulated by something water channel or something. Then for brain function, these water channels are very important. For the first time, water channel was identified by using Xenopus laves oocyte, kaeru no tamago. Xenopus laves oocyte, and the 28 kilodalton protein found in human red blood cell was confirmed very fast water permeation pore that is eventually named aquaporin 1. And in 2003, my collaborator Peter Agre win a Nobel Prize. Okay, then from them, intensive study appeared, and then all life forms on the earth have water channels. For example, in our body, as you will see, 13 water channels were identified and are involved in numerous physiological processes. And intensive study about aquaporin 1 gave us puzzling questions. Aquaporin 1 can permeate 3 billion 10 to times 9 water molecule can permeate in a second per channel. only one channel can permeate 3 billion water molecule, very effective water permeation channel. But this channel exclude any ions and even proton. Proton's regulation is crucially important because pH condition for any cells are very important because signal transduction and proliferation and survival of cells are regulated by pH, means proton. Then water channel have to exclude proton, even when permeate very fastly. Then, for example, proton gradient is used to synthesize ATP, which is energy source of our body. For example, in our brain, ion pump use this ATP and then continuously pumping NAK ATPase is pumping and then make a gradient and then ion channel can work. Then we can think in our brain. But exclusion or inhibition of proton permeation is not so easy to understand because for changing pH condition, hydronium ion does not need from one side to the other, but just exchanging hydrogen bonding partner through so-called water wire, and effectively a proton can move from one side to the other. To block proton permeation, hydrogen bonding should be separated, isolated. But in such case, energy barrier rise up, and then impossible to permeate 3 billion water molecule in a second. So how our inner body water channel can achieve such amazing function? For answering these questions, we have to analyze structure. We did. And it's turned out we had thought water channel have to be simple because, as I mentioned, any life forms on the Earth, including even primitive life forms, have water channels. So we had thought water channel have to have a simple structure, but not actually very complicated. For example, loop B and loop E come into inside and form short helices from almost perfectly conserved NPA sequence. NPA means asparagine proline alanine sequence. Asparagine in this NPA motif back bonded to NH group, are you okay? NH group by carbonyl group, then start short helices. As a result, two amide groups are stick into inside the channel, and as you will see, stick into the inside channel, and then at that height, this height, narrow, very narrow constriction is formed. Diameter is just 3 angstrom. Water is 2.8 angstrom in diameter, then just one water molecule can pass through such a narrow constriction. Whereas any hydrated ions and solutes cannot permeate, then block. So strict water selectivity is achieved by forming narrow constriction, as you can see here. But at the same time, because of complex holding, forms are this kind of structural feature, molecular dynamics simulation showing you, and red ball means oxygen is face to all oxygen molecule, water oxygen is face to your side, means NPA side. electrostatic field formed by two short helices forced to orient water-oxygen phase to NPS side. And then smoothly form a hydrogen bond with two amide groups. Such hydrogen bond formation renders them, in turn, this hydrogen atom to perpendicular to the channel axis. Then such orientation renders them too far away from adjacent molecule, and then hydrogen bond is isolated here. Then proton permeation could be blocked here at the center of narrow constriction. Then this structure was very good, but the resolution was just 3.8 angstrom. We could observe no water molecule in the channel, but unfortunately, we could analyze another water channel aquaporin 0 at 1.9 angstrom resolution. At such high resolution, we could see the aromatic rings of phenylalanine and tyrosine with holes. And very importantly, inside water channel, we could see a water molecule clearly. And this arrangement is very similar with cartoon which I made based on aquaporin 1 structure. And then this structure strongly support hydrogen bond isolation mechanism, selectivity mechanism is okay. But unfortunately, this structure of aquaporin 0 at 1.9 angstrom resolution was closed state. Then actually only 3 water molecules could be observed in the channel. To explain very fast water permeation, then very difficult to understand from this structure. Then we have to check the observe fast water permeation channel, aquaporin 4. Aquaporin 4 is predominant water channel in our brain and expressed in endophyte of astrocyte and ependymal cells and glial lamella of hypothalamus. You'll see here. Then we could confirm aquaporin 4 can very quickly permeate water molecule. We analyze structure by using electron crystallography at 2.8 angstrom, and 8 water molecules are clearly discriminated in the channel. The density at 1.8 angstrom, smaller number is better. So high resolution analysis by X-ray crystallography were, however, something blurred. Then this kind of counterintuitive notion could be attributed to surrounding atmosphere. This is very strange phenomena because higher resolution blurred, lower resolution clearly discriminated. To explain this reason might be care about surrounding atmosphere when we try to analyze structure. By electron crystallography, we could analyze membrane protein inside lipid bilayer. Lipid bilayer known to have a characteristic dielectric constant. Distribution of characteristic dielectric constant enforces the strong helical dipole moment. But in case of such as X-ray crystallography, dissolved and disappeared lipid bilayer will give a small helical dipole moment. In cooperation with such arrangement of strong helical dipole moment, carbonyl groups are arranged at the opposite side and then act as hydrogen bonding partner for water molecule inside the channel, otherwise perfectly hydrophobic narrow channel. Then these arrangements can be, and also amide groups of perfectly conserved amide groups of asparagine is a lower the energy barrier and can permeate very rapidly water molecule, water channel pass through such a narrow path. And then when we see the distance of here is 3.9 angstrom and 3.8 angstrom, by which no hydrogen bond formed, then safely hydrogen bond is isolated. Proton permeation can be blocked here. So our cell can be overcome pH change and survive. This kind of helical dipole moment arrangement can be observed in any ion channels ever analyzed. So these results suggest helical dipole moment looks very important. Okay, so actually in our, for example, glial lamella of hypothalamus, a large amount of aquaporin 4 are expressed, and then looks like aquaporin 4 adhere glial lamella, means cell and cell is adhere. And our analysis by two-dimensional crystal, double-layered two-dimensional crystal, teach us how to adsorb two layers. You'll see here 3-10 helix interact. But one may suspect this kind of interaction is caused by artificial crystallization. Then by using L cell, fibroblast cell, which has no endogenous cell adhesive molecule, then we confirmed aquaporin 1 have no cell adhesive function, but aquaporin 4 has. And quite interesting thing is this arrangement of water channel is not straightly aligned, but shifted. Then when in brain, water osmotic pressure change, that the water permeate, then this adhered membrane might be separate. And this system might be good for osmosensor because our neural cell is very weak to pressure change. So we have to very sensitively sense osmotic pressure. Then this kind of function, cell adhesive function and channel function, both come to one molecule, aquaporin 4, and then aquaporin 4 can regulate complex things. For example, we don't know in detail, but suicide manic depressive, can you understand? 自殺する躁うつ病の方の his brain has a large amount of aquaporin 4. So we want to know why such a water channel related with such a higher order brain function. We are getting somehow a model, and it's too early to explain in here, but we hope we will be able to clearly understand such mechanism if possible. Thank you for your patience. Hear me out, my poor English, but I would like to add a few words. The bacteriological structure was a nice collaboration with these peoples, and aquaporin 0 was collaborated with Thomas Balz, who used to be in my lab when he was a student in Basel, Switzerland. And also, aquaporin 1 was a nice collaboration with Andreas Engel and Peter Agre. And aquaporin 4 work was mainly achieved by Yoko Hiroaki and other people. And this is just computer simulation, molecular dynamics calculation passed through aquaporin 1 water channel. Thank you again for your patience to hear me out.
Thank you very much, Professor Fujiyoshi. So now we move on to the question and answer session. So please line up behind the microphone in the middle row.
So I really enjoyed your speech. Your speech was very powerful, and I'm interested in the 13 kinds of AQP. And do you say so? All the creatures on the Earth have the same kind of AQP?
Structure is very similar, but an essential part is some part is different, but an essential part is same.
So like the dipole helix.
Yeah.
Oh.
To show the helices arranged inside channel and form. But this the constriction is 3 angstrom, but the
Yeah.
other channel has 4 angstrom. Such one can permeate glycerol, not only water. So, but exclude ions. If ion permeate, then we will be crazy.
Oh.
Because doesn't work ion channel. So we are thinking by using ion channels. So water channel has to perfectly exclude any ions.
Okay.
Okay.
And another ion channel such as sodium channels or calcium channels
Yeah.
also have to be also have to exclude other
Ions
ions.
shows ion selectivity. Sodium channel have to exclude potassium or calcium, other ions, but only sodium can permeate.
Okay. So so let's think for another ion channels and so for example about calcium channel. Are there many kinds of channels such as
Yeah.
water?
Yes, there are many variety of channels.
Oh.
So, and also still one potassium selectivity was clearly explained by Roderick McKinnon, who got Nobel Prize because of he explained the selectivity. But remaining things, sodium selectivity, calcium selectivity is not clearly explained yet. No one can explain. So please come to this field and study the ion selectivity. You may have a, I don't know, but maybe have a chance to get special prize.
Oh, I see. So sorry.
Yeah.
This is
So-
my
so,
last question.
the-
Sorry.
Yeah.
Yeah. So why do you think the aquaporin or other ion channels need so much variety? So not
Yeah,
only one.
because many biological processes require such variety of channels.
Okay. Thank you very much.
So many people can want to ask a question. So one question per person, please.
Okay.
So next one, please.
Hello. I enjoy your speech, and then I have one question.
Yes.
Uh, is there any molecules that can through the AQP except water?
Yes, as I mentioned, for example, aquaporin 3 can permeate glycerol, a little bit larger than water, but surely can be permeated. So that glycerol channel is very important for beautiful, especially women. So, but even men. So yeah. And also curing such cut or such harmed skin. So
Thank you.
yeah, thank you.
Thank you very much. So next question, please.
I enjoyed your speech, although it was quite difficult for me. But what I want to ask is, you've discovered that the 80 or more percent of brain is consisted of water. That is the higher number than other parts of our bodies. Is this right?
You mean
Our body is consist of majority of the water, 60 or 70%, but brain
Yes.
is the higher, has a higher number of percent
Yeah.
of water. So why does the brain need so much water contents than other of our body.
Our element, you mean?
Yes.
Yeah, so maybe life forms on the Earth maybe start to form in the sea or water. Then I'm not so much. It's you are talking about brain.
Brain.
Brain, I don't know, but brain neural cell have to float on water.
So the reason why the
I
water
don't know.
brain has a more water content is not so clear yet.
No,
That's
no,
right.
not so good. At least I don't know.
Oh,
Thank you.
thank you.
Thank you. So next question, please.
You can speak.
So I have one question.
Yeah.
So actually, does immunity become higher by drinking the water?
Immunity?
Immunity.
I think drinking water is good, but yeah. So and then I don't know immune system, but maybe a medical doctor will answer. But I'm not a suitable person for answering the question. But something water is definitely important for our body, and then you need water, drinking water. So I recommend you will drink water a lot. But yeah, and when you drink alcohol, you shouldn't now, but later you will drink. Take care. Expression of aquaporin 4 in our brain is increased. So quite dangerous brain edema appeared when you have an injury on brain. So take care.
Thank you.
Yeah, thank you.
Thank you. So next question, please.
And thank you for your wonderful presentation.
Thank you.
So is it possible to create artificial AQPs and increase them in human bodies?
Wow. And I'm not clearly understand your aim, but we have a artificial expression system, so gene technique can produce aquaporins expressed in anywhere. But when your question is make something water channel by polymer or the other materials, it's very hard. I don't know. I have just now no idea. If possible, water selective, strict water selective filter, effective filter, something like aquaporins, then very useful for changing, sea water change to pure water. So may need, but I don't think it's easy. It's surely hard.
Thank you so much.
Yeah.
Thank you.
Thank you.
So next question, please.
Nice to meet you.
Yeah. Nice to meet you.
Do you think that what you should do to the house of a high school student for being a researcher?
Oh, it's good question, and maybe you will keep or you will find even simple question and quite interesting question for you. And if you can keep long years, then that is most important thing for growing to good researcher, I think. Please keep find your question or very interesting things, and then keep it.
Thank you.
Yeah, thank you.
Thank you. Next question, please.
Hello, I'm interested in the relationship between the brain and the mind. I'd like to ask you a question about this. Why do some men identify themselves as women?
Yeah, yes, it's important question, but I'm not perfectly good level brain scientist, so I'm not. So it's very difficult to answer from me. So could you ask someone, a suitable person? I can introduce someone. So, for example, Ikegaya in University of Tokyo, and he will be answer. He is my friend, a very young but very talented person. So you can ask Ikegaya. I recommend him. Yeah.
Thank you very
Yeah.
much.
Thank you.
Thank you. So next question, please.
Hi, I was really interested in what you said about the potential relationship between suicides and water channels. And so I wanted to ask if there are other effects of water and water channels on the psychology of human beings.
Yeah, it's a good question. So I have a model, and then yeah, I prepared something slide. Maybe someone will ask such questions. So I am expecting, because that is a very hypothetical model which I made. So we couldn't prove yet. So then I have to this is my hypothetical model, very hypothetical. But you know, in our brain, when you know, I'm a man. If I want to hug to beautiful woman, try to, and then my cerebrum will block such terrible behavior. But they have brain, and then try to regulate by themselves or himself, and it's very bad. But I want to show you in cerebellum. We know cerebellum. Here is cerebellum. When you ride on bicycle, so when try to falling off, and then there is a feedback. For example, when I touch fingers, two fingers, and this is error, then we can touch. That is, feedback neural system was clearly observed. But in cerebellum, we have no such neural network. Then should have. And at astrocyte, proton might be the key molecule. Okay, key molecule. So key ion. So proton may feedback to at a peer outermost cortex, and then feedback to neural cell. But when something or drink person express large amount of water molecule, and such patient have a large amount of aquaporin 4, then block the proton permeation. Should be then the patient cannot stop to suicide, kill himself or herself. That is my hypothesis. But we have to prove. Yeah.
Thank you so much.
Thank you.
Hello.
Next question, please.
Nice to meet you.
I have a question about AQP.
Mm-hmm.
Does that difference affect the character of a person who has a lot of AQP and who hasn't?
Yeah, it's a good question. There are regulations. As I mentioned, in case of drinking alcohol, large amount of aquaporin 4 is expressed when you didn't drink alcohol. So then there are such regulations. And also aquaporin 2, when you feel thirsty, then aquaporin 2 come out to your renal duct, and then water suck into your body. So there are dynamic regulation of water channels.
Thank you.
Thank you.
Thank you. Time is so limited, so this last question, please.
Yeah, okay.
Hi, I'm Willem Van Dam. I was just wondering, so you've been figuring out the different structures of the aquaporins. How could we use that information to track brain activity, or different ways that we can apply it?
Yeah, it's a good question. For example, as I mentioned, when you drink alcohol and then got injury, and then cytotoxic brain edema will kill. Then we try to find inhibitor of water channel based on structure. So we are developing a drug to cure or block such dangerous injury. So that is application for us to analyze structure.
Thank you.
Yeah, thank you.
Thank you. So we'll like to conclude the question and answer session. So thank you very much, Professor Fujiyoshi. Big hand to him.